आप सभी प्रबुद्ध जनों को मेरा नमस्कार। आज ज्ञान गंगा कार्यक्रम के 5वें दिन मैं डॉक्टर नितिका सिंह आप सभी का हार्दिक स्वागत करती हूँ। आज के कार्यक्रम की थीम है रिमोट सेंसिंग एंड बायोमेट्रिक। तो हम आज का सेशन प्रारंभ करते हैं। Now we are going to start our session. The first resource person of today is Dr. Dharmendra Singh. It's my pleasure to introduce Dr. Dharmendra Singh. Currently, Dr. Singh is working as Assistant Scientist, Environment Ecology, Haryana Space Application Center, Hisar. Sir is having more than 10 years of experience in remote sensing applications for environment or ecology. Sir has served as at various organizations of national importance like Forestry Survey of India, Dehradun, Space Application Center, Ahmedabad, ISRO, FRI, etc. Sir has 25 publications in peer-reviewed national as well as international journals and conferences. Sir have completed project on IWMP and air quality monitoring funded by ISRO and environment department respectively. Sir have guided more than 10 students at PG and PhD level. Today, Sir is going to deliver his talk on remote sensing of vegetation. I welcome you, sir. You can start your presentation. Dr. Dharmendra Singh. Sir, you can start your presentation.
So, good morning to all of you. Is my presentation is visible?
Yes, sir.
I'm audible also?
Yes, sir.
Okay. Thank you so much for introducing me in this short-term training program under Gyan Ganga for assistant and associate professors of botany. And just I'm making it full screen, my presentation. So, okay. So from okay. So today's theme of presentation is basically remote sensing of vegetation, and basically this particular presentation is of my interest because I am working in this domain since last 10 years, and this is my privilege to present some of the experiences that I have gone through while working as well as doing PhD in the same domain. This whole presentation would be focused on the applications of remote sensing for vegetation science, how remote sensing is applicable, and slowly we will know the remote sensing as a science, and then how what is the basic principles of remote sensing that governs the vegetation science, governed by the vegetation science. And after that we will get some of the applications that are very popular in today's geospatial world. So from now while starting this I'm starting this. So first when we saw the remote sensing as a science, so what is remote sensing basically? So it is a process of acquiring information about an object without physically contacting it. So this is a common remote sensing, which we don't take it as a subject when we go for earth science or remote sensing of earth environment. So for remote sensing of earth environment, it is basically uh, recording and measuring of electromagnetic energy reflected from or emitted from a planet's surface or earth's surface, you can say. So when we talk about remote sensing, it is related to all the remote sensing that we do, whether it is planetary remote sensing or it is Earth environment remote sensing. Everything is included in remote sensing per se. But when we talk about the remote sensing of Earth environment, it is basically measuring and recording of electromagnetic energy that is actually reflected from the Earth surface features or emitted from the Earth surface feature. Basically, remote sensing includes some of the components. That is, one is your sensor. You can see in the figure that some of the sensor are in the space. Basically, sensors are the devices which sense the energy, you can say, and which sense the target that we interested in. So this sensor basically captures the reflected energy from the Earth surface. This is Earth, and this reflected energy is sensed by the sensor. And this sensor sense these energy in many of the electromagnetic domains. That may be visible, that may be mid infrared or thermal infrared or microwave infrared. Some of the remote sensing applications are now also approaching to the ultraviolet, but rarely seen in the X rays or gamma rays, cosmic rays. So wherever we saw in recently, there is a visible remote sensing. This small part of the electromagnetic range, electromagnetic spectrum. Uh, okay, so this is visible in blue and green, but this range micrometer to 400 nanometer to 700 nanometer, and this is called actually photosynthetically radiation. So when we started remote sensing, it was the vision one purpose, but nowadays using this technology, okay, am I visible and means
Sir, you are audible. Please share your screen.
it is already shared. Share. Okay, so it is low. I click visible. So I just talking about the visible remote thing that we practice. I told you the visible includes the blue range, green range, and red is purple range. Ranges from 400 to 700 nano. and some of the movements in near infrared also we are doing remote sensing in the noise. So overall, if I say remote sensing, it is science in which we measure or records the electromagnetic energy that जी 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 ठीक है सर. So now I think it is connected well.
Yes, sir. Please share screen.
Oh, sorry for the inconvenience. Actually, okay. So here it is okay. Now come to the principle of remote sensing. What exactly happens when we saw remote sensing as a science, and how remote sensing is actually done when it is the matter of vegetation science. So there are basically three principles of remote sensing. One is detection and discrimination of object, means detection and recording of electromagnetic radiation reflected from the object or emitted from the object. Second is different earth surface objects reflect different amount of EMR or electromagnetic radiation in different wavebands. And the third one is amount of EMR is basically depends on physical and chemical as well as structural properties of the material. So if you see, there are three principles. One is detection and discrimination in remote sensing is directly means recording or detection or recording of EMR that is reflected from the object. The second one is the uh, different surfaces that have different response towards the EMR, and you can see in this figure actually here. So this particular spectral signature is basically vegetation signature, and this is a vigorous vegetation. So it has a spectral response that is very unique as compared to water as well as soil. So in earth surface, we have basically three kinds of object or material. One is water, another is vegetation, and third one is soil. So all these materials reflect different energy in different bands or different electromagnetic radiation. So if you see in X direction, this is wavelength. You may assume this as a band also, and then reflectance percentage. And this is basically how much percentage is reflected from a particular surface. So if you see the water is actually, this notation is looking somewhat different. This one is soil. Uh, this one is soil, and it reflects maximum in NIR region, while less in the visible as well as blue and green, red and green. While for water, it reflects maximum in green, and then it is almost absorbed in NIR region. And then if you see vegetation, it has a very unique signature. So I told there are three principles. One is measurement of light. The second principle is different object reflects different amount of energy. So these are different objects or materials, and they are reflecting different amount of energy. But this amount is dependent on the physical, chemical, and structural property of the material. So if you see this particular spectrum, here you can see some of the absorptions, and here some amounts is also there. So this is 0.8%, 47%, or this is 20%. So if you see, this percentage is basically representing the moisture percentage in soil. So when the moisture is high, the absorption is more. When the moisture is less, then absorption is less. So this particular reflectance is only for the dry soil. And this is different wet shades of the soil. And that's what I told. The amount of material. So amount is varying in this particular soil. Amount of moisture is varying in the particular soil, and that's why different EMR is reflected. So this is what the principle uh, tells us about the vegetation or remote sensing as a science. So this is what you can see, some of the um, FCC, where you can see the fires are there, and then this is satellite, uh, which takes this kind of image. The satellites moves in an orbit around the Earth, and that orbit may range from 500 to 900 kilometer above the surface, because this is a polar satellite. So there are two types of satellites. One is polar satellite, another is geostationary satellite. So geostationary satellites are actually at the height of 36,000 kilometer, while polar satellites rounds the earth uh, within 700 or 800, or you can say 600 kilometer above the surface. So different heights are there, various satellites are there, and they are at different heights, and they have different band combination. They have different number of bands, and based on this principle, they are categorized as geostationary as well as polar satellites. So this is basically rounding. A polar satellite is revolving around the Earth, and when it is geostationary, it would have revolved like this with the speed of Earth. So this is the principle of remote sensing. Come to the next, how the remote sensing is actually done. What is the mechanism of remote sensing? So if you see, I told you the polar satellites, they revolve from pole to pole, while geostationary satellite, they are at equator generally with some angle, and they revolve with the speed of Earth. So they looks like stationary. So in remote sensing, what basically happens, the sun radiation comes or approach towards the Earth's surface. Here it interact with atmospheric clouds as well as different materials that exist in atmosphere. And some of the radiation which comes from atmospheric window hit the Earth's surface features like urban or soil or vegetation, and it reflects back to the sensor. Then sensor record this reflected energy, and this reflected energy is converted into such type of digital images, where you can see this green means red is basically vegetation, this open cyan color is basically the fallow land, and this dark cyan is basically your uh, vegetation settlement. So this is what the remote sensing mechanism is. So once we get such type of images, we bring this to some remote sensing image processing softwares, and here we basically analyze these images to get meaningful information for the management of vegetation and other natural resources. So here you can see the yellow one is incoming radiation, it is called. Then reflected radiation is green one, red one, and then emitted radiation is basically blue one. So emitted is what? Once this light comes to the Earth's surface, the Earth's surface features does not reflect everything into the atmosphere. They absorb something. and hence there is no source of the radiation, then this absorbed energy get emitted, because the temperature, or you can say temperature, flows from high to low environment, you can say. So what basically we do in general remote sensing or optical remote sensing, what we focus, actually we focus on this reflected EMR, electromagnetic radiation, that is red portion of that. When this reflected portion is absorbed by this particular sensor, it absorbs vegetation in such a manner, soil in this manner, and water in this manner. So there are different bands, and when these band combinations are made, such type of images are obtained. So this is the mechanism of remote sensing. So I told you the reflected energy is basically your target that you go through, or you would like to have some interest on that, because this gives you fair chances of science through remote sensing. So reflected energy is basically called reflectance. So what reflectance is? This is a ratio of output divided by input, or you can say outgoing radiation divided by incoming radiation, or that is coming from your sun. So this is called radiance basically, and this output is called the measurement of surface complicated by atmosphere. So this is basically ratio of your reflect means ratio of incoming and outgoing radiation is called reflectance. Input solar radiation basically uh, is a source of energy for passive optical remote sensing. So when the source of energy is sun, then this is called passive remote sensing. And when the source is spacecraft, or you can say sensor itself, it is called active remote sensing or active system. And such type of systems are basically your radar. So how many type of radiation reflectances are there? So mirror reflection, or specular reflection, or diffuse reflection. So this is basically your interest that is required for your remote sensing, optical remote sensing purposes. While in SAR, you have backscatter values. So this is similar to the reflectance, but different terms is used in backscatter, where maximum energy returned towards the sensor, which is sent through sensor itself. Come to the another important aspect of remote sensing, that is called atmospheric window. So what I told, the incoming sun radiation that comes from the sun is basically does not come as such. There are small windows in the atmosphere which allows the radiation to pass through, and that's why when we do remote sensing, or we design some sensor for remote sensing, we require the knowledge of atmospheric windows. So what atmospheric windows are basically? These are some of the special or specific regions of electromagnetic radiation where there is no absorption from atmospheric constituents. So maximum things get passed through this, and these small portions are called atmospheric window, and these are transparent to the sun radiation. So there are specific regions like 400 to 700 nanometer, or 400 to uh, uh, 2500, you can say 2500 nanometer range is basically atmospheric window. There are some of the satellite or sensors which does not require these kinds of things, like radars, which have a full coverage of the things that, but basically works in the microwave domain. So when we talk about the microwave domain, it is free from the atmospheric interventions or atmospheric turbulences. So it is all weather remote sensing we do. So this is what the atmospheric window is. Come to the remote sensing data and resolutions, how we define. So there are four type of resolutions. One is the spatial resolution, another is spectral resolution, the radiometric resolution, and temporal resolution. So spatial resolution means the clarity of your image, means as much as small size pixels you have. This is basically called pixel. When you take a data, a remote sensing or satellite data, it is a pixelated form, and each pixel you can say is a cell, and this image is made up of rows and columns. Rows and columns are divided into cells, and cells are associated with x, y, and dn. What is x, y, and dn is x is latitude, y is longitude, and dn is your digital number. Similarly, when you see this image as a zoom, or without zoom, then you can see these kinds of images. If you zoom this, you will see this kind of observations, but pixelated forms. But see, this is a high resolution, high spatial resolution image, and this one is your low resolution images. So when spatial resolution is high, you can see a clear picture where the pixels are small in size, while a low resolution image, low spatial resolution image, where the pixel size is more, and you have less clarity of the all features that exist. Come to the spectral resolution that you can see here. This is a multispectral where you have only three sections of electromagnetic energy, or you can say bands 1, 2, 3. This is called blue, green, and red. This is multispectral, or you can say low spectral resolution. But when you come to the high spectral resolution, then you can see multiple bands in blue region, multiple bands in green region, and multiple bands in red region. So in between, there are yellow and cyans also there. So this is what, when you have more number of bands, and you have narrow spectral ranges, this is called the satellite or remote sensing data which you get is called high spectral resolution data. But when you have few number of bands, it's called low spectral resolution data. Come to the radiometric resolution. So radiometric resolution basically, see, so it is measured in the bit. So if it is 1 bit or 2 bit or 4 bit or 6 bit. So see, this is nothing but the number of values in a particular band, you can say range of your values in a particular band. So if you take blue band, so if it is a 2-bit resolution, then 2 to the power 4. So 4 shades would be there: black, then gray, and a small gray, and then white. So there are 4 shades of this. So 4 values are possible only. So this is what the radiometric resolution. While temporal resolution is frequency of acquiring the data. Means a satellite which is revolving around the Earth, what is its capability to capture the data? Means how many days it takes to cover the same land surface or same area of the Earth surface. So this particular time is called the temporal resolution. Come to some of the important satellites that are revolving around the Earth and giving immense number of supports in terms of remote sensing of vegetation, as well as in many other domains and today's governance also. So remote sensing satellites like Landsat, this is a NASA satellite, and this has a long series. Currently Landsat 8, it is called OLI, is working. Then Sentinel 2 and 1 is also there. So this is the highest resolution that is available from European Space Agency, and it is free of cost. Anybody can download the data and use this data. Then the another one is Resource-Sat 2A. This is an ISRO satellite. It has various sensors like LIS-3, LIS-4. So these actually covers the Earth, whole Earth, means list recovers in 13 to 14 days nowadays, and list 4 in 23 days. So this is highest resolution in multispectral data that ISRO have. Then another satellite from NASA, which is providing data free of cost. So this is MODIS, called a moderate resolution imaging spectroradiometer, and this gives various or n number of scientific products that can be used for vegetation science, like LEI or surface temperature or air temperature, or you can say vegetation condition or vegetation health, phenology, burnt area. So n number of things it is providing. The MODIS, which is freely available to access to everyone. This data is also free from Bhuvan, but only few. Uh, this is an old data. The new data you can find from Landsat, Sentinel, and MODIS also. So these two satellites are from NASA. This one is from ESA, and this one is from ISRO. So these are few satellites. There are n number of satellites. More than 4,000 satellites are there currently working, and I have just presented four to concise this presentation, and these are basically free of cost. There is no cost associated with these data. They are provided free of cost. So anybody can use this data. Anybody can download this data. Come to the application sections where remote sensing is applied. So here I have mentioned some of other satellites like Spot, GeoEye, RadarSat, WorldView, TerraSAR, or OrView, or Iconos, or Pleiades, or Sentinel, Eros, MODIS, QuickWord, Landsat, RapidEye, then InviSat. In spite of this, we have this ISRO have INSAT, Kalpana. So these are the satellites of communication and weather satellites, INSAT and Kalpana. While some of the satellites mentioned here, many of them are for, you can say, Earth remote sensing or Earth science, and some of them like InviSat, or you can say this INSAT or Kalpana, they are weather satellites. So we can use the remote sensing in image classification or multi-temporal analysis or orthophoto generation or digital aviation model generation or crop classification or many other applications are there for different environment or for different areas such as land use or land cover, environment planning, transportation planning. These are the some of the applications of optical as well as radar remote sensing. Transport planning, urban planning, infrastructure, mapping, survey, mining, agriculture, energy, hydrology, digital aviation model, geology, disaster, risk assessment. So recently in disaster section, if you observe, the ISRO has identified the exact reason which happens and what happens in Uttarakhand recent disaster, and this was the remote sensing satellite data, which actually gives the clue how this havoc happens, how did this disaster happens. So basically, temporal analysis was done, and they identified landslide as a major reason for this havoc or this disaster, which happens, and many of the people, means 2,000 or more than 3,000, 300, 200 or 300 people died in this disaster. So what I mean to say, there are n number of applications of remote sensing. But since today's application is focused only for the vegetation, I am just focusing for vegetation only. So forestry applications are there. So what remote sensing application for vegetation is? Uh, Basically, the applications can be divided into two sections. One is qualitative remote sensing for vegetation, and another is quantitative remote sensing for the vegetation. So I will present some of the example for qualitative remote sensing of vegetation and some of associated with the quantitative remote sensing. So qualitative means this is a physical, you can say physical appearance, or you can say quality of vegetation. This is qualitative remote sensing. When you say the quantity means how much area has gone, or how much carbon is present, or how much nitrogen is present in your. So when you talk about the amount, we say for biomass, these are the quantitative remote sensing. When you quantify the parameters, so there are n number of parameters in vegetation, like the vegetation scientist or botanist like to assess the type of vegetation that is present, or you can say the species identification means what species is present in a particular forest, because in forest this is not accessible to all actually. When we have remote sensing data and we have technique, so we can identify what species is distributed where. So once we have a species, we can do habitat type of analysis, means what type of habitat this particular species is forming. Say, for example, if you have information or a species level map for ficus. So ficus is a keystone species, you can say, and this serves as a shelter for many of the birds and animals that reside in the wood. So once you have a map of this, you can do some habitat analysis. Where should this particular species would be? So this is what the quality. So quality is related to the physical, or you can say physical appearance of the vegetation or quality of the vegetation. While quantity is related when you quantify something using the remote sensing data. So how the remote sensing of quality and quantity is done? So see, I told you this particular signature, how the vegetation, a healthy vegetation reflects, and why these reflections are. So if you see, this is wavelength and this is reflectance. In each wavelength, there is various percentage of reflectance. So I told the principle of remote sensing is amount of radiation reflected is governed by the physical, chemical, and structural parameters of the vegetation. So see this particular section that ranges from zero point 0.4 to zero point 0.7 micrometer. It is called leaf pigment section. And when it is 0.7 to 1.3 micrometer, it is called cell structure region. And 1.4 to 2.5, it is called water content region. So if a vegetation, if you would like to analyze the vegetation chlorophyll or vegetation other pigments, you need to have these kinds of bands or these spectral regions, measurement in these spectral regions. If you would like to analyze the cell structure, whether it is shrink or whether it is flaccid, or you can say what type of cells are there, how much layers are there. So you need this particular domain that is called an IR region. And when you would like to analyze the water content of vegetation, you need 1.4 to 2.5 meter micrometer region. So this is called infrared region, this is called short wave infrared, and this is called visible region. So for leaf pigment, you need visible spectrum. For cell structure, you need near infrared region, and for water content, you need short wave infrared region. So this is what the vegetation respond towards the EMR. So once you know what spectral region reflects what, you can use a particular section for the assessment of various things like leaf pigment or cell structure or water content. So leaf pigment, what you get from leaf pigment is basically health of the vegetation, or you can quantify uh, the yield of vegetation, you can say. So overall, uh, this is the science behind your vegetation remote sensing of vegetation. So absorption in these regions, if there is more absorption, then there would be more chlorophyll. And if there is less absorption, there would be no chlorophyll. Similarly, if there is more absorption, less reflectance, there would be more water in the vegetation. And if water stress is there, then this absorption will be less. So similar science you can apply in remote sensing of vegetation. See the qualitative remote sensing, we can assess the burnt area. So where are these burnt areas? So if I put an natural color composite, burnt area looks in blue color. So where blue color you are seeing, it is burnt area, and you can easily digitize these things, and you can further do quantitative remote sensing, means how much burnt area happened. So once you identify where the burning has gone or where the burning has occurred, then it is a qualitative remote sensing. But when you do an area assessment, how much area is burnt, that is quantitative remote sensing. Come to the another one, vegetation health assessment. So you can do the vegetation health or crop health condition. So here you can see high health, and then less green is medium, and then yellow one is poor, and then these things are non-vegetative regions. A temporal analysis from 1997 to 2017, you can see many of the area that was not an agriculture or known crop converted to the crop, and accordingly their health had changed. Though it is converted into crop, but there is still some of the region where there is not a good health. While here this one is a canal, so along the canal you have very good health of you can say vegetation, because there is plenty of water. So and this can be obtained from the uh, vegetation indices like NDVI. And this is the false color composite. When you do a band combination, or you use different spectral bands to get an image, it is something looks like this. So you can see there is a lot of non-crop areas, and visually you can also see there this has been changed now into different land use, and it is agriculture only. So and it is visible in the health index also. So this is how you can analyze the vegetation health, or you can assess on a regular basis. Further, you can do a qualitative analysis like the canopy density assessment, which is a premier work that is done by the Forest Survey of India. This was actually my earlier organization, so I have some of the information from there. So see, from ground, if you see, this is something very dense forest, then this is moderately dense forest, this is an open forest, and this one is scrub. So these four kind of densities are basically practiced or mapped by the Forest Survey of India on a biennial cycle basis, and they are basically assessing this forest, I think, early 80s, and now at present they have various organization or centers uh, around the India, and they do this kind of analysis on biennial cycle basis. So in every 2 year they map the canopy density for whole India using the list 3 data, such type of data. So here you can see the dark color is they basically identify this as very dense forest, and very dense forest means the canopy cover is greater than 70%. While less dark, like such kind of patches where, and this one they call this as medium dense forest, where the canopy density ranges from 40% to 70%. And then this light red color, they identify this as open forest, and the canopy density ranges from 10 to 40%. Along with this, they also map the potential forest, that is called scrub, where the canopy density is less than 10%. So there is canopy density you can see, and you can correlate with this. So this is a scrub, this is a moderately dense forest, or MDF, or this is VDF, or this is open forest. So what I mean to say, they all are using the remote sensing data, and this remote sensing data is giving qualitative information about the forests of India, and that's why canopy density is a qualitative information. And this canopy density is assessed through your remote sensing data, that is list 3. So every year they do, and this report you can find on their site, FSI website. So anybody can go there and use these reports. This is a very nice report and exhaustive information about the vegetation. So those who are fond of doing remote sensing of vegetation, they must go through the site and just download some of the reports and just read it out or get some information. So state wise, district wise, forest cover information is available, and they have more than I think 14-15 cycles. So immense number of information can be obtained. And since early stage of remote sensing, now they are advancing their technologies, and they are using this remote sensing for canopy density assessment. They called it forest cover map, so FCM. So this forest cover map is basically represents the canopy density, and canopy density is when it is 10 to 40% is open, when 40 to 70% is medium dense, and when it is greater than 70% is dense forest. Come to the another one, qualitative remote sensing, forest type assessment. This is again a work of Forest Survey of India. So what they used to prepare this forest type map is basically the vegetation indices, the topography, the climate and rainfall, elevation, and the base data of champion and set forest type classification. So champion and set, I think in 1969, they have given a detailed forest types of India, and they use this base data as a reference point, and mapped the forest types of whole India. So here you can say tropical evergreen, or tropical moist evergreen, or tropical moist deciduous, and then temperate forest. So many type of almost 16 type subtype forest they have mapped, and 102 subtype forests are mapped by Forest Survey of India. So this data is also maybe available if requested. So this is how the remote sensing works. So remote sensing can be used for vegetation, indice generation. Remote sensing can be used for topography. Remote sensing can be used for rainfall. Remote sensing can be used for elevation data. What you need a literature data. So once you have literature data, you have all these inputs from remote sensing. Combining all these things, you can get forest type map. And this is what qualitative remote sensing is. Means this is again telling you quality of your vegetation. So what type of forest is. Come to another application of qualitative remote sensing, that is called leaf area index. So leaf area index is what basically it is one-sided leaf area per unit ground area. So for leaf area index assessment, you need a ground data. So ground data can be collected through many ways. There are instruments like PAR, and you can also collect some HemiView photographs. This is a picture of HemiView photographs, and then you can analyze this in some software. You will get the ground NDVI and ground leaf area index. Then at the same location, you can obtain information or measurement from your satellite. You can have a correlation between your satellite measurement and the LAI which you measured from the ground. Once you have this correlation, uh, you can apply this relationship on this image, and you will get this kind of LAI ranges. This is ranging from 2.5 to 5.5. So the red one is high NDVI region, and this black or blue region is basically low NDVI region. So you can see these are low regions, and this is basically residing near to the rivers. So it is obvious there would be less LAI if there is river. So this is visible. So you're fitting a model and then obtaining the leaf area index. Similarly, you can apply this to various days or temporal analysis of LAI. So here it is less LAI. Once it is increasing the day when they are showing something 208 day, and then at 213 days it is full of green, which shows the high LAI. So overall, this is remote sensing based information that you can get uh, as a qualitative property of your vegetation. So the assessment of leaf area index or biomass, these are called basically biophysical parameters. So when you go through literature, you may find somebody tells this as biophysical parameter retrieval, and inside the literature they mention the leaf area index, biomass, and etc. And there are another parameter that is biochemical parameter. So in this they measure various kind of biochemicals like leaf nitrogen or leaf chlorophyll. So these are the quantitative, you can say, quantitative remote sensing. Once you assess the leaf area index through remote sensing, it has n number of applications, because many of the geophysical and biophysical models require leaf area index. Many of the models are there. If you would like to do some erosion analysis, or if you would like to do some hydrological analysis, or biochemical analysis, or energy and mass flow analysis. So everywhere this particular property of the vegetation is required. So yield analysis also is being done using this. So what I mean to say, remote sensing is a science which gives you this data, this information, and this you can use in a number of models. Come to the next one, that is again qualitative remote sensing. So using remote sensing, you can distinguish species type also. So this is a PhD work that was done by me only. So this is my PhD work. So where I have discriminated 16 type of species in part of Doon Valley. So first we have generated spectral signatures, a spectral library for various species. So 1, 2, 3, 4, there the name is mentioned. And after this spectral library, we get this hyperspectral data. And then this hyperspectral data was classified. So spectral angle mapper classification. And this kind of map was obtained. So here you can see this is top one. So this is mostly oak forest, and this is citrus forest. Some of the places they have a pinus forest also, and then the lower portion have the sal forest. This is also sal forest. This is a pine forest of FRI Dehradun, and then here it is subtropical forest of Mohania. So a clear-cut observation you can, or clear-cut information you can see here. They see how these two patches are looks like here. These are sal forest till here, and this small portion, the small gap you can say it is Bohnia forest, and then apart from this, this is oak forest. So overall, what I mean to say, the quality of forest is assessed, or quality of vegetation you can assess through remote sensing. In quality, you can assess the leaf area index, you can assess the forest type, you can assess the forest density, you can assess the vegetation species even. And the same you can apply for the crop also. Come to the another section, that is quantitative remote sensing. So quantitative remote sensing, what exactly gives you amount of vegetation? So any change in cover over time, or bearing property of soil and vegetation or canopy cover gives you the quantitative information. So see, if it is bare soil, and if it is healthy soil, and if it is senescence field. So here bare soil, full cover and senescence, you can easily quantify how much amount of vegetation or what is the area of this particular vegetation is. So this amount gives you the quantity, and that's why it is called quantitative remote sensing. Come to the another one, that is biomass assessment. So biomass is basically is a very important parameter for global climate change and global carbon assessment. So if you would like to know your forest or vegetation is absorbing carbon or it is a source of carbon, you need to have information about the biomass. So once you have biomass information, you can assess the carbon, total carbon in your forest, and a temporal analysis can give you amount of carbon or changes in the carbon amount. And that's what this information you can use in climate change models. So biomass assessment is basically done in similar fashion as leaf area index being done. So once first we go to the field and we collect data from the field, then we come to the lab and identify the locations, and then we get satellite measurements from these locations. We have or we do some relationship or establish relationship between biomass and spectral signature, and then this is inverted to the satellite, and this kind of biomass information we can get. See, you can see this red portion or this yellowish portion is having less biomass, while the blue and less blue portion is representing high biomass. So what I mean to say, this relation you can use for your biomass assessment. So this is what the biomass assessment. So because once you do the biomass quantification, that's what is quantitative remote sensing, and you can quantify the biomass, you can quantify the carbon from this biomass, and you can assess the value, total value of the carbon that is assessed. So once you have carbon, so you can approach this carbon to the carbon credit market also. So starting from a small point, that is satellite remote sensing, you are converting this satellite data into biomass, and then biomass into carbon, then carbon is bring to the economy. So this is how everything is connected with each other, and what is you and how you can apply this for a uh, vegetation science, you can say how vegetation is performing or how vegetation is supporting your economy. Performance of vegetation can also be assessed. So if there are some unhealthy vegetation or there are disease in the vegetation, it will reflect less. And once it reflects less, then there would be less biomass. And if less biomass is there, you can easily identify and go to these patches, and then in these patches, you can do some management activities. So there I have a very good example of forest disease management that happened in early 90s. Actually, Hoplo Ceramics was insect which makes hole in the sal trees. So due to this, a large patch of sal forest get degraded, or you can say dried. So, and this was observed by the Indian Institute of Remote Sensing scientists, and they identified. They went to the place and they saw there is the disease. So they informed the forest officers. They went to this forest officer, uh, make some strategies based on the plan that was made through remote sensing data. They appoint some of the local peoples to kill these hoplocerambics, and by doing so, they have killed all the disease insects, and now there is a very good sal forest exists in that area. So this is what the remote sensing actually gives. Quantitative information gives you. Again, I told you the quantitative information, you can assess the carbon stock. So for carbon stock, you need satellite measurement. From satellite measurement, you can get vegetation index. You have some models that can measure the biomass, and this biomass can be converted into the carbon, and carbon can be presented into your map. So this is Amazon forest, and here the carbon density is high in these areas near to the rivers, and as the forest is reaching far away from the rivers, it is showing less carbon density. Here, see, very less carbon density is there, or low, zero. There is no forest. Similarly, we can assess the chlorophyll also. So once chlorophyll is assessed, you can use this data for the yield assessment. So a high correlation exists between your chlorophyll and yield. So, and there are a number of models are also established, and they are reported in literature. So you can go through literature, you can obtain these informations. Then how we actually do the chlorophyll assessment? We have satellite measurement. Similarly, we develop vegetation indices using these satellite measurements. We have some models. We assess the leaf chlorophyll first, and then canopy chlorophyll first. So this is a PhD work that was done by me during my PhD. So I have done this canopy chlorophyll assessment of the same reason which I showed you earlier for species mapping. So this is what the citrus and the oak forests are there. So they have high chlorophyll content. While sal forest, this was a deciduous time, or you can say leaf senescence time. So very less uh, canopy chlorophyll was obtained. But here it is uh, river actually. So along the river, there are some vegetation which are healthy, and they they have high chlorophyll content. So wherever the rivers are there, it is high chlorophyll content. Uh, this is reason of high chlorophyll content. So the data which we used was hyperspectral data, but optical data can also be used in a similar fashion. So one by one, you process through these steps, you can get the chlorophyll. Once you can get the chlorophyll, you can assess the yield. And once you have yield assessment, you can give it to the government offices for further planning. So overall, this yield is quantity. You can say how much kilogram per hectare is going to be produced this year. So this information is basically used by the government for making new shelters or new storehouses. And this early information, if they know, they don't import actually. They know that what the species is and how much they are going to yield. They do this analysis. and they use this information for the management purpose. Then come to the canopy nitrogen assessment. So here in similar fashion, so you can see satellite measurement can be used for the generation of vegetation indices, and these vegetation indices can put in some models to get the leaf nitrogen, and this can be converted into canopy nitrogen. So from traveling to from canopy to the from leaf to the canopy is a cumbersome task actually, and for this we have some ready-to transfer models like ProSail or Sail, but these are beyond the scope of the current presentation, so everything is not presented here, but this is a simple flow that gives you an idea that how the canopy nitrogen is assessed actually. This is again my PhD work. Then come to the another one. This is lignin assessment. So here you can see some of the areas and basically the river sides alongside the river. They have high chlorophyll, lignin, and the temperate vegetation. The pine forest you can see. They again have high lignin. So because they are high lignin content vegetation types. You all know would be knowing actually. Because pine leaves when they fall on the surface, earth surface, they due to lignin only, they don't allow everything to grow actually, and that's what the quantitative remote sensing helps you on the ecosystem management, you can say. So overall, if you see, you can use for quantitative and qualitative remote sensing for the management of ecosystem or vegetation, you can say, vegetation ecosystem. Again, this is a recent work that was done over the Western Haryana region. So this is an ecosystem service assessment, you can say. So in this, you need to have satellite measurements like land use, land cover. Then you have vegetation indices, and these vegetation indices can be used into the models, and which models further gives you the ecosystem service values. So see, the distribution of ecosystem service values. So red patches are basically the ecosystem services that is provided by your particular land use, and this is varying actually from this USD per year. So this is 6,855 USD minimum, starting from 0 to this, and maximum this is 1190.4717 USD per hectare or per year. So and for per patch also. So these things you can see, and in temporal analysis you can assess whether your ecosystem services are increasing or decreasing. So these are another aspect of quantitative remote sensing. So if I conclude the whole session in a nutshell for remote sensing of vegetation. So remote sensing of vegetation are of two types: quantitative remote sensing and qualitative remote sensing. Qualitative remote sensing includes the assessment of canopy density, forest type, species, leaf area index, and quantitative remote sensing includes the assessment of biomass, carbon, then ecosystem services, and then forest area change. So see this forest cover change assessment you can see. So this is a previous year image and this is the new year image. So here this open land converted to the forest, while there is density change. So there is open forest and there is medium dense forest. Similarly, there are negative changes that can be assessed through this remote sensing technique, and this is actually this was a solar system that is near to the Rajasthan. Some portion is there, and it is converted into the solar panel system. The forest is converted. In chain detection, you can see this is an earlier image, 1975. So once you go next in 1986, how these houses are made in this area. Again in 1992, this changes. Similarly, in agriculture science, you can go for crop health assessment, then water content assessment, then crop classification that I told you. But overall, I would like to give some information about data providing agencies like Airbus, Copernicus, NASA, and then Bhuvan, JAXA, then Earth Data, NOAA. These are some of the famous organizations that provide you data of remote sensing. These are some of the links you can use to download data free of cost to do remote sensing of vegetation, of course. So this is from my side. I would like to conclude from this last slide that you all are welcome to have some queries if you have. And yes, thank you.
Thank you so much, sir. Now, I would like to invite Dr. Manoj Kumar Rawat, co-convener of the program, to deliver vote of thanks to Dr. Dharmendra Singh. Dr. Manoj Kumar Rawat.
थैंक यू निकिता मैडम. आयुक्तालय कॉलेज शिक्षा राजस्थान जयपुर व राजकीय महाविद्यालय बूंदी के वनस्पति शास्त्र विषय विभाग के द्वारा आयोजित ज्ञान गंगा कार्यक्रम में उपस्थित सभी प्रतिभागीगण, मैं धन्यवाद ज्ञापित करता हूं डॉक्टर धर्मेंद्र सिंह, सहायक वैज्ञानिक, हरियाणा स्पेस एप्लीकेशन सेंटर हरियाणा, जिन्होंने पर्यावरण अध्ययन की महत्वपूर्ण शाखा रिमोट सेंसिंग के बारे में सभी प्रतिभागी गण के ज्ञान में वृद्धि हुई. आपने रिमोट सेंसिंग के प्रिंसिपल, मैकेनिज्म, रिमोट सेंसिंग सैटेलाइट्स और इनका एप्लीकेशन, क्वालिटेटिव एंड क्वांटिटेटिव एप्लीकेशन के बारे में आपने विस्तार से हम सभी को लाभान्वित किया. धन्यवाद.
Thank you, sir. Thank you so much.
थैंक यू सो मच सर. सभी प्रतिभागियों के लिए एक जानकारी है, कृपया सभी ध्यान दें. जो प्रतिभागी स्वैच्छिक रूप से कल वैलिडिटी फंक्शन में अपना फीडबैक देना चाहते हैं, कृपया वे चैट बॉक्स में अपना नाम लिखें. आप सभी को कल 3 से 4 मिनट के लिए बोलने का मौका दिया जाएगा. तो जो प्रतिभागी स्वैच्छिक रूप से फीडबैक देना चाहते हैं, वे कृपया अपना नाम चैट बॉक्स में लिखें. दूसरी जानकारी ये है कि इस प्रशिक्षण के लिए जिन संकाय सदस्यों को नामित किया गया था, उसमें से 7 संकाय सदस्य ऐसे हैं जिन्होंने अभी तक रजिस्ट्रेशन नहीं किया है. उनके नाम मैं बोलने जा रही हूँ, कृपया ध्यान दीजिए. अजय शर्मा, राजकीय महाविद्यालय डीडवाना. पुरुषोत्तम लाल, राजकीय महाविद्यालय धौलपुर. ऋषिकेश मीणा, राजकीय महाविद्यालय झालावाड़. जितेंद्र एस राठौड़, राजकीय महाविद्यालय खेरवाड़ा. सुरेश कुमार, राजकीय महाविद्यालय कोटपुतली एवं शर्मिला, राजकीय महाविद्यालय नीम का थाना. आप सभी से अनुरोध है, यदि आप सभी इस ज्ञान गंगा कार्यक्रम को अटेंड कर रहे हैं तो कृपया अपना रजिस्ट्रेशन कंप्लीट कीजिए. धन्यवाद. Now it's time to introduce our next eminent speaker. I feel honored to introduce Dr. Harish. Dr. Harish is currently working as Assistant Professor Botany at Mohanlal Sukhadia University, Udaipur. Dr. Harish has 15 years of teaching and research experience. He has qualified NET and equivalent exams 5 times. He was awarded PhD in 2007. He was later awarded with prestigious Dr. D.S. Kothari postdoctoral fellowship. He has published more than 30 research papers in international as well as national reputed journals with total impact factor of 53. He has guided one PhD student and 4 are currently pursuing PhD. He is currently running a research project funded by DST New Delhi worth of rupees 50.51 lakhs. He is also having one joint research project with one professor in France with total cost of 1.60 crores. Today, he is going to discuss concepts of statistical analysis. I welcome you, sir. Please deliver your talk. Dr. Harish.
Thank you, Nitika. And first of all, I would like to thank the organizers for inviting me in this really wonderful program, Gyan Ganga. Special thanks to Dilip ji also. Let me share my screen. Okay, so in this talk, I am going to discuss about some of the concepts that are popular and that are useful for the statistical analysis of the raw data. So in this talk, I'm going to discuss about the basics of the correlation and how it is different from the regression. And we will discuss the simple linear regression. Then I'll be talking about the two types of error that we commit during experimentation and the concept of the degree of freedom. What are the hypotheses, null hypothesis and alternate hypothesis? What are the probability of errors and level of significance and analysis of variance? And if time permits, then we will of course go for the practical demo using some software like Excel and Visio. So what is experimental design? Let's start with the basic concept. So suppose we have a population and we want to estimate, let's say biodiversity we want to estimate. So we don't go for the estimation of the biodiversity for the entire population. We take some sample like quadrate, we take measure the quadrate size and then we take the biodiversity measurement in that particular quadrate. So that is known as sample. So whenever we are analyzing the any parameter, so we estimate the parameter in a particular sample and then we estimate or extrapolate the results of the sample for the entire population. So in every book you will find that there is one statistical term that is used for sample and the another is for the population. So suppose we are designing one experiment, we want to analyze the effect of any compound, like for example zebralic acid. We want to test the effect of zebralic acid on the plant height. So during experimentation we need to have one control and treatment. So control means we have not given any kind of zebralic acid in a plant, and in treatment we can have a different concentration of the zebralic acid. And to nullify the effect of genotype in the plant height, so we can have a different replicates. So why we are taking replicates? Because to nullify the experimental error or variation within the genotypes. So minimum 3 replicates are required for any sound experimental design, because suppose replicate 1 in control, the response is like plant height is increases, and in replicate 2 the plant height does not increases or it remains the same. So to conclude whether the plant height increases or does not increases, we need to have a value of the replicate 3. So that is why we are taking the 3 replicates. Then the response is what we are going to estimate. It may be plant height, suppose in this case, or it may be some other parameter, like for example, protein we are estimating. So we can take the protein value as a response. So response is anything that we are going to estimate. So in experimental design, we have a control, we have treatment, we have replicates, and we have a response. So these 4 things are essential that we need to clarify in our experimental design. And also we need to understand that we should frame a question in our experiment. What kind of question we are going to answer by doing that experiment. If we are clear with the question, that suppose whether the gibberellic acid is going to affect the plant height or not. So this is the question. So if you can frame a question, then only you can design the experiment accordingly, and then you can estimate the respective parameter to answer that particular question. So suppose we want to estimate the protein value. So we take the leaf leaf of the plant, and then we crush the leaf and they add some chemicals. Suppose Lowry method we are using. So we crush the leaf and then we develop the color. So in the leaf, we don't know what is the protein value. So to estimate the protein value in the leaf, we need to have a known value, and that known value is known as standard. And standard is a series of concentration. It starts from 0 concentration, which is known as a blank, and then it goes on increasing, like 1 milligram, 2 milligram, 3 milligram of the protein concentration. So there is a known concentration of the protein, and that known concentration of the protein starts from the 0, and that series is known as standard. And the blank is that component which has a 0 concentration of the protein. So there is a difference between the blank and the control. Control is part of your experiment, in which of course you are going to estimate the protein also, in control also. So that to estimate the protein in control also we need standard. So blank is the part of the standard and control is the part of your experiment. And next thing is that what is calibration? So suppose we are going to estimate the OD of the standard. So we are using some instrument like spectrophotometer. So we will measure the OD of the standard, which starts from the blank, and then we will keep on noting down the OD. So whether the spectrophotometer is measuring the correct concentration, correct OD or not, that we have to determine. So in blank, we have to set the value 0. So setting the value of blank as a 0 is called as a calibration. So calibration is something which is required for every instrument. For example, in pH meter, suppose we don't know whether the pH meter is accurate or not. So we need 3 buffer solution, pH 4, 7 and 9.2. And first, these buffer solution are known with their pH. So they are accurate. So if suppose the pH meter is showing 3.5 pH for a buffer solution which is actually having a 4 pH. So we need to rotate the knob on the pH meter to make it accurate. So that rotation of the knob of the pH meter to give the reading of the 4 pH as a 4 is called as calibration. So calibration is essential for any experimentation if you are using any instrument. So these are some of the basic concepts that we have highlighted in this particular slide. So sample is only a portion of an entire population, and we estimate the parameter in the sample, and then we extrapolate the result for the entire population. In experiment, we have to decide the control, treatment, and replicates, and whatever the response that we are going to use, it may be a protein or it may be sugar or chlorophyll, whatever it may be, we need to have a standard. A new standard is essential because for example, today we are estimating the protein. So today the temperature, humidity, and voltage in the electricity may be different from the standard which was done earlier. So every day, whenever you are measuring any parameter, you need to have a new standard, and you need to measure the value in the standard. That then only you can have a correct absolute value for the response also. So and to need you need to make auto zero for the blank, that is known as calibration. So let's start with our different statistical analysis that we use in our experiments. So first is the correlation. So what is correlation? You know, there are two types of analysis. One is univariate analysis and another is bivariate analysis. So suppose we are estimating the mean, median or mode. So such type of analysis are known as univariate analysis. While if we are establishing the relationship between the two variables, so such analysis is known as bivariate analysis. So using the bivariate analysis, we can measure the strength between the two variables, that is known as correlation, and we can predict the value of one variable using the value of another variable, that is known as regression. So there is a difference between the correlation and the regression. So correlation basically gives us the strength between the two variables, how much relationship is strong between the two variables. And prediction using one variable, predicting the value of another variable is known as regression. So there are different types of correlation. First type is based upon how many variables you are analyzing. So if we are using two variables, that is known as simple correlation. And if you are using more than three variables, that is known as multiple correlation. And suppose the experiment was performed in with many parameters like yield, rainfall, fertilizer, all these things you have measured. But at a time you are analyzing only the two variables, but the response is based upon many variables, and you are analyzing two variables. So such type of experiment is known as such type of analysis is known as partial correlation. Then depending on the direction of the change in the two variables, there are three types of correlation. Suppose increase in one variable results in increase in another variable. So such type of correlation is known as positive correlation. Suppose there is an increase in variable of increase in size of the variable in x variable results in decrease in another variable. Such type of relationship is known as negative correlation. Then if there is no correlation and that values are changing at random, so such type of relationship is known as no correlation. Then there is a third type of the correlation. So suppose change in one variable results in equal change in another variable. So there is a constant change in between the two variables. The ratio of the change between the two variables is constant. Such type of relationship is known as linear correlation. And suppose the change in one variable does not bring the same amount of change in another variable. The ratio between the two variable change is changing. So we have a curvilinear or non-linear relationship. So basically there are three types of correlation depending upon the number of variable, the direction of the variable, and the ratio of the two variables, change in two variables. So the first scientist who gave the formula for the quantitative estimation of the correlation, which is known as coefficient of correlation, is given by the Carl Pearson. This formula is quite complicated, but if you closely look at this formula, you can point out that there is x variable, which is independent. There is y variable, which is dependent variable, and there is third variable xy. So multiplication of the independent and dependent variable. So by dividing this formula into three parts, you can easily memorize this formula, and you need to add the root also. So linear correlation coefficient, which is known as small r, ranges from -1 to +1. So -1 represents the perfect negative correlation, and +1 represents the perfect positive correlation, and 0 value indicates that there is no correlation. So basically, depending upon the value, away from the 0 indicates the strong relationship between the two variables. Away from 0 means either in the negative direction or in the positive direction. So suppose value is a 0.9. So there is a strong positive correlation, or value is minus 0.9. That means there is a strong negative correlation. So strength of the relationship can be estimated using the correlation. So how we declare the result for the correlation? Suppose the our calculated value or computed R value is greater than the tabular value. The standard tables are available in the statistical books. So if R computed is greater than the R tabular at 5%, that means the relationship is significant, and relationship exists by the 95% probability. If the R computed is greater than the R tabular at 1%, that means the relationship is highly significant, and relationship exists by 99% probability, and only 1% there is a error. If R computed is less than R tabular at 5%, that means there is no correlation between the two variables. So this table shows some data. You have plant height on X value. These are independent values, and depending upon the plant height, you have a number of flowers. So number of flower is Y. So this is a dependent value dependent upon the X value. So we have a X and Y values, plant height and number of flower, and we want to estimate whether the two variables have any correlation or not. So is there any correlation between the plant height and the number of flower? So we can calculate this correlation by manually as well as using statistical software. So first we will learn manual calculation. So you have X square, you need to calculate the X square, then Y square, then XY. So X square is X multiplied by X, Y square is Y multiplied by Y, and then multiply X and Y. So in this way, you need to calculate these square and multiplication value. Then at the end of the table, you need to add the summation of entire value. So you have a summation of X, you have summation of X, Y, summation of X square, summation of Y square, and summation of multiplication of XY. So these value we have calculated using simple calculator. So now we know the formula for the Pearson's coefficient of correlation. So we can place our value X square and Y square and XY, and using this solving this formula, we can estimate the value of our correlation, that is 0.908. That means yes, there is a significant correlation between the two variable, plant height and number of flower. So this is how we calculate the correlation using simple calculator. So let's understand the concept of regression. So regression, as we have discussed earlier, it predict the value of one variable using the value of another variable. So prediction is possible. So what is regression basically? So we have one independent variable which is known as a cause, and one dependent variable which is known as Y. So independent variable results a change in the dependent variable. So depending upon how many cause and how many effect are there in our experiment, there are different types of regression. So suppose we have a one cause and one effect. So such type of regression analysis is known as simple regression. And suppose there are multiple cause for one effect. So suppose plant height as well as sunlight. There are two factors which brings about change in the number of flower. So we have a multiple regression. So this is type 1 of classification, simple and multiple regression. Another type of classification is depending upon the change in the ratio of the two variables. If the ratio between the two variables is constant throughout the entire range, such type of relationship is known as linear regression. And if the ratio is not constant, such type of relationship is known as linear relationship or non-linear relationship. So these are different types of regression. Simple regression can have a linear relationship and non-linear relationship. Multiple regression also can have a linear and non-linear or curvilinear relationship. So let's understand the difference between the correlation and regression, again using simple diagram. So we have a independent variable here as a x and a dependent variable y on the y-axis. So suppose what correlation tell us whether the two variables are have any relationship or not. So strength of the relationship is revealed by the correlation. So suppose we have a different sets of data. So there is one set which shows yes, there is a positive correlation between the x and y. There is another set which says the same thing, that yes, there is a positive correlation between the x and the y. And there is one more data set, third set data set, which says yes, there is a positive correlation between the x and y. So all these three data set represented by green, blue and orange line says that there is a positive correlation between the two variables. But how the relationship between the x and y? So change in x, how much change in x results in change in y? So that we can predict using the regression. So if we extrapolate the green line on the y-axis, we bring about quantification of the intercept value, and we can estimate the slope value also. So if we can estimate the a value and we can estimate the b value also, then we can predict the change in one variable results using the change in another variable. So regression basically tell us that whether the line is like this or line is like this, or correlation is like this. So what is the slope between the different data set that can be estimated using the regression. So simple linear regression equation is known as y is equal to a plus bx. So a is your intercept value, b is your slope value, x is your independent variable, and y is your dependent variable. So if we know the a value, we know the b value, and we know the x value, so you can calculate the y value. Or if we know the y, a and b, you can calculate the x value. So this is the formula for calculating the a and b value using simple calculator. So a can be calculated by mean value of the y and b and mean value of the x. And this is the formula for calculation of the b value. So suppose in our previous experiment, we have estimated the OD of the standard, which means we know the concentration of the different test tube having 0 milligram, 1 milligram, 2 milligram protein. So we know the OD of the known concentration of the protein. So using the OD of the known concentration of the protein, we can plot a standard curve. And using that standard curve, we can bring about information about the A and B. That means we can find out the regression equation. So now we have the y, a, b and x value with us. So suppose now we are taking the leaf sample from the plant and we are measuring the OD. We don't know what is the protein level. So using the OD value, you can estimate the protein value, or using the so this is the formula. So we can estimate the protein value using the OD of the leaf sample and subtracting the intercept and dividing it by the slope value. So this is how the prediction is possible in the regression. So let's use the same example for calculation of the b value and a value. So we have x and y, x square, y square and xy. So we have estimated these summation values. So using this summation value, we can calculate the b value. So b value is 0.98, and using b value, you can calculate the a value. So a value, that means intercept value, is 3.54. So if we know the intercept, we know the slope value, you can, and we know the y value, that means OD. So you can calculate the protein value in any unknown sample. So this is the manual calculation of the regression. So next concept is different types of error. So what is hypothesis? Hypothesis is any state which needs more evidence. So or in our experiment or in our research work, we made some statements, and those statements need to be supported by some sort of evidence. So suppose we have in research we have found out one medicine which is beneficial for the coronavirus. So suppose in research we have found that medicine is a beneficial, or suppose in research we have found a medicine which is harmful. But in reality scenario, so research result can be different from the reality scenario. So in reality, suppose we have two possible outcome. Medicine may be beneficial, or medicine may actually be harmful. So there are two scenario in the reality, and there are two scenario in the research. So suppose let's draw the Punnett square diagram. So we have a reality scenario and research scenario. Suppose in reality, in research, we have found a medicine which is beneficial. In research, it is a beneficial, and in reality also, the medicine is beneficial. So we have drawn a correct conclusion, and we can release the medicine. But let's understand that in reality, in research, we have found a medicine which is harmful, and in reality also it is a harmful. So in that scenario, we have identified a medicine which is having a more side effect, and that is that we can stop it from releasing. So there are two correct conclusions that we have drawn. But suppose we have an error in research. So in research, we found that a medicine is harmful. So in research, the medicine is harmful, but in reality scenario, in actual scenario, the medicine was actually beneficial. So we have committed a type 1 error. And in research, we have found a medicine which is beneficial, but in reality, it is a harmful. So we have committed a type 2 error, and type 2 error is more dangerous. So let's understand this type of error using COVID-19 test. So suppose we have tested one person, and we found it either negative or positive. And in reality scenario also, we have a two possible outcome. It may be negative or it may be positive. So we can draw a conclusion, same conclusion. So suppose in COVID-19 test, the person is negative, and in reality also the person is negative. So we have drawn a correct conclusion. In COVID-19 test, we have found a person positive, and in reality also the person is positive. So we have drawn a correct conclusion. But there can be some error. So in COVID-19 test, we have found a person positive, but in reality, the person is actually negative. So if we have observed a person positive, we can guide him that you remain isolated for a few days. So in that particular situation, we have committed type 1 error. So type 1 error is not so dangerous. But suppose in COVID-19 test, we have observed a person negative. So in test result is showing that person is negative, but actually in real scenario, the person is positive. That means we have committed a type 2 error, and type 2 error is more dangerous. So our main aim is to reduce both the error, and in general, it is more risky or dangerous to accept a false hypothesis. We cannot accept a false hypothesis. And it is less risky to reject a true hypothesis. The type 1 error is less risky, and we can reject the true hypothesis, although it may later on it may be find out that there was a type 1 error during previous experiment. So it consumes the time and energy and money also. So type 1 error is delaying the actual research result, and type 2 error is more dangerous and more risky. So we need to have experiment in such a way that both the type of error reduces. So this is all about the two types of error. Let us understand the one more concept, that is degree of freedom. So degree of freedom is in statistical analysis, we always measure the degree of freedom, and we don't know what actually that mean. So suppose this is the standard book definition of the degree of freedom, that number of values that can be assigned to statistical distribution. So by this definition, it is very difficult to understand what is the degree of freedom. So let's understand this degree of freedom using some simple example. So suppose we have 7 different hats and we want to wear a different hat every day of the week. So on first day, suppose Monday, we have a choice of 7 different hat and we have selected this particular hat. So we have a choice on first day, that is on Monday, we have a choice of 7 different hat and we have selected this particular hat. So on second day, that is on Tuesday, we are left with 6 choices only because we cannot wear the same hat. We have, the condition is that we have to wear a different hat every day. So on the Tuesday, we have a choice of 6 different hat and we have selected this particular hat. On Wednesday also, we have a choice of 5 different hats. On Thursday also, we have a choice. And on Friday also, we have a choice. And on Saturday also, 6th day of the week, we have choice. So we have choice of 2. As soon as we select the hat for the 6th day, we are left with no choice on the 7th day. So actually, we have a 7 minus 1, that is 6 days of hat freedom. So degree of freedom is 7 minus 1. So let's understand the degree of freedom using some sort of statistical data. So suppose we have a data set of 10 values. There are 10 different values and the average of these 10 values is 3.5. That means the summation of these 10 value should be 35. And we want to design or we want to pick up the data for these 10 value. So we have a freedom or we have a liberty to select any data for the first value. So first value you can select any value. Second value also you can select any value. Up to 9th value you can select anything. And as soon as you select the 9th value, you are left with no choice. That is on 10th day you have to select a value so that our total of the 10 value is 35. So on the 10th value, you have no choice, but you are limited by the choice selected on the first 9 values. So all the 10 value have a summation of 35 and mean value of 3.5. So 10th value cannot vary. It must be a very specific number. So let's understand this using an example. Suppose on the first value you have selected 34. Second value you have selected minus 8.3. Third value you have selected minus 37. Fourth 92, minus 92. Likewise, you have selected different values and 9th value you have selected is 99. So as soon as you selected the 99, the 10th value is now very specific. 10th value is now 61.3. So if you don't select the 61.3, the total will not be 35. So all total of all these 10 values should be 35. Then only you can have a answer of 35 and an average of 3.5. So you have a 9 freedom, 9 choices, but the 10th value is fixed. Same is with this example. Suppose we have selected 0.1 value on the first value, 0.2 on the second, 0.3 on third value, likewise 0.9 on the 9th value. So as soon as you pick up the 0.9, the 10th value is becomes very specific, and that is 30.5 to make the total 35. So therefore you have 10 minus 1, that is 9 degree of freedom. So it doesn't matter whether what is your sample size or what is your mean, or the sample, the last value of the sample cannot vary. That means you end up with n minus 1 degree of freedom. So n is your any number of size. So this is the basic concept of degree of freedom. So if we can define again the degree of freedom. Degree of freedom is the number of value that are free to vary. So 10th value is not free to vary. So our degree of freedom is 9 in our previous example. So this is the concept of degree of freedom. Next concept is types of hypothesis. So hypothesis is any statement which is based on insufficient evidence. So it can be true or it can be false. When we need to test our hypothesis using experiment, and we need to prove that whether the statement is correct or statement is wrong. So let us understand the types of hypothesis using one example. So suppose we have given two types of treatment, two types of water to a plant, and we want to estimate the biomass of the plant after 30 days of the treatment. So we, on one plant we have given tap water, and on another plant we have given distilled water, and we want to analyze whether the two types of water have any significant effect on the plant growth or not. So there are two possible outcomes. So first is, no, there is no difference between the two types of water. Whether you give the tap water or whether you give the distilled water, plant health will remain the same, or plant biomass will remain the same. Such type of hypothesis is known as null hypothesis, which states that there is no difference between the two types of treatment. Another alternative hypothesis is that yes, there is a significant difference between the two types of treatment. Such type of hypothesis is known as alternative hypothesis. So researcher want to prove the alternative hypothesis by rejecting the null hypothesis. So why we are using the null hypothesis in alternative hypothesis? Because it is much easier. It is much easier to demonstrate that statement is false. Because proving something false require only one counterexample, while proving something true require many evidence. So it is difficult to prove alternate hypothesis. It is easier to reject null hypothesis. For this reason, when we conduct an experiment, we design a null hypothesis. The alternate hypothesis is a claim, is a statement that is made by the researcher. So alternate hypothesis is a claim that researcher actually tried to prove. However, they prove the alternate hypothesis by proving that null hypothesis is false. So they prove that null hypothesis is false, but they claim that alternate hypothesis is true. So if the null hypothesis is false, then opposite, alternate hypothesis must be true. So that is why we design, or we give two statements. Null hypothesis, that means there is no significant difference, and alternate hypothesis means yes, there is a significant difference between the two types of treatment. So what is the level of significance? So suppose we have one experiment and we have estimated some plant height or protein value or whatever you want to estimate, and we repeat the experiment again, and we repeat it 100 times, and out of the 100 repetition we got the same result for more than 95 times. So that means our results are same. Suppose we have given one compound and we want to estimate the plant height after 30 days, and we measure that yes, after 30 days the plant height is increases, but the same result we should get for more than 95 times. So suppose we got the result for more than 95 times, that means the error is less than 5%. Or if we get the result for less than 95 times, that means our error is more than 5%. So in science, you cannot have a more than 5% of error. So less than 5% of error is scientifically valid. More than 5% of error is scientifically invalid. So, and it is difficult to perform an experiment 100 times. So we have statistical analysis so that we can calculate the probability of error. So this error we can calculate. using the error, we can estimate whether our experiment are scientifically valid or not. So there are three parameters in the level of significance. First is probability of error. So probability of error you can represent by two different ways, either by percentage value. So percentage value ranges from 0 to 100, or you can represent the probability of error using coefficient which ranges from 0 to 1. So 0.5 or 0.6, 0.7, in this way you can have a coefficient value. Level of significance means whether the control and the treatment have a difference or not. And if there is a difference, what is the significance of the difference, whether it is a significant or highly significant difference. So in this way, you can correlate the probability of error and level of significance. So there is a non-significant difference because there is a more than 5% error, or there is a significant difference because there is a less than 5% error. So if the null hypothesis is having less than 5%, results for the null hypothesis is less than 5%, and for alternate hypothesis we are getting for more than 95%. That means our statistical, there is a significant difference between the control and the treatment, and that significant difference is represented by level of significance. And same, if we go for the sampling from the population. So we can have 100 times resampling from the population, and if 95 times we get the same result from the resampling, so we can have a confidence interval, which means yes, our sample are true representative of the population. If we get more than 5% error in the sampling, that means our sampling is not actual representation of the population. So this is how the three parameters are linked together. These are the different levels of the significance. Probability of error, if more than 5% error is there, that means there is a non-significant relationship between the control and the treatment. That means control and treatment are same because we are getting more than 5% error, and this is represented by NS symbol, and the confidence level is less than 95. If the probability of error is less than 5, that means our results are significant, and we denote this result using single star, and confidence interval is more than 95. If we are getting less than 1% error, that means our results are highly significant, and we denote this result using 2 star. If we get 1 error out of 1000 experiment, that means our probability of error is 0.1%. Such result is known as highly significant, and we denote such result using 3 star. So this is the different types of level of significance. And further, statistically significant should not be confused with the commonly used word like important or something big, because statistical significance means our samples are true representative of the population, and we have a variation between the treatment and the control. That means statistically we have a sound evidence that yes, there is a difference between the treatment and the control, and our alternate hypothesis is correct. But this significant word is look like it is very important or it is something very big. So news report always highlight this, that our results are significant. So we should not confuse our results. The statistical significant and the important word are two different and separate things. So let's understand the concept of ANOVA. So suppose we have designed our experiment and we want to understand the effect of X compound on the plant growth. So we have replicate value, 3 replicate value, and we have a different treatment level. And this is the plant height response. So we can estimate that whether there is a difference between the control and the treatment or not. So ANOVA basically tell us that whether there is any difference exists between the control and the treatment, or there is no difference between the control and the treatment. So how this ANOVA measure the difference between the control and the treatment? So first it measure the variation between the replicates. So this variation, there should not be any variation because all the replicates are under the same treatment level. So we have not added any X compound here in this. So suppose this is a pot experiment. In this pot, we have not added anything in the same here also and here also. So there is no difference. So this difference is due to genetic variation between the plant. So if the plant height is 10 or 11 or 13, it is because of the genetic difference between the plant, because all of the rest of the factors or parameters that can influence the plant height are nullified or removed in our experiment. And here we have given a logical X compound, the concentration of the X compound. We know the concentration of the X compound in treatment 1, 2, and 3, and in control also. So we can estimate the variation between these different parameters. And the variation between replicate and variation between these different treatment, we can estimate. The ratio between these two variation is known as ANOVA. So ANOVA is basically variation, explained variation. We know the concentration. These are the explained variation. And we don't know the variation, why there are variation, because the treatment level is same in all the replicates. So these are known as unexplained variation, or unexplained variation is due to genetic difference between the plant. So if there is a variation here or in these three replicates, and same level of variation are also here also, so our F value will be near to 1. So there is non-significant difference. But if the explained variation are more than the unexplained variation, that means F value will keep on increasing. And more the F value means, yes, there is a significant difference between the treatment and the control. So ANOVA basically measure the variation between the explained variation, variance, and unexplained variation. Or we can simply say that it measure the ratio between the group variability and within the group variability. Here in this experiment, we can state two types of hypothesis. Null hypothesis, which means that all treatment are giving same result. There is no difference between the treatment. Alternate hypothesis says that no, there is a difference between the treatment and the control. So there are two statements, null hypothesis and alternate hypothesis. So ANOVA basically tell us whether there is a difference between the control or not. And if there is a difference, what is the level of significance, whether it is a significant or highly significant difference. So if there is no difference, we denote such result using non-significant NS wording. If there is a significant difference, that means we denote our result using single star. And if there is a highly significant difference, we denote such result using 2 star. Or if there is a highly significant difference, we denote using 3 star. So this is the history of the ANOVA. First of all, William Govt developed this t-test. In the t-test, you can measure only 2 variables. So control and treatment. You can compare the mean value of control and the treatment, and it can give you the answer that whether the control and treatment are same or not. So at a time in a t-test, you can compare only 2 variables. But in ANOVA, you can have more than 3 samples. So ANOVA was developed by Ronald Fisher, and because of the surname Fisher, this test is also known as F test. So there are 2 types of ANOVA in general. If we are calculating only 1 variable, so such type of ANOVA is known as one-way ANOVA. And suppose we are giving yield, we are estimating the yield of the agricultural crop, and we are having 2 parameters, rainfall and fertilizer. So rainfall and fertilizer, 2 parameters we are analyzing. So such type of ANOVA is known as two-way ANOVA. In this particular workshop, we are going to discuss only one-way ANOVA. So ANOVA you can calculate either by manual simple calculator, or you can calculate using statistical software. So in this slide, first we will discuss about the manual calculation of the ANOVA, which is a very long process, and then at later part we will have a demo on software, which is very easy using one click button. So manual calculation requires 7 different steps. So first step is you need to have a you need to calculate the treatment total, block total, and treatment mean. Then you need to determine the degree of freedom. Then you need to calculate the correction factor and sum of square. Then mean square. Then you can compute the F value, dividing the mean square by error mean square. You can compute the F value, and then you can obtain the tabular F value, and you can declare the result. So these are the 7 steps. So let's understand each step one by one. So suppose we have a such type of experiment where there are 4 different types of replicate value. Block means replicate 1, replicate 2, replicate 3, and replicate 4. And we have given different treatment. So we have 5 different treatment level: 2, 4, 6, 8, and 10. And this is the response. Now we need to calculate the block total, treatment total, mean treatment, and total grand mean. So this is the red line, surrounded data is what we get. And this is the block total which we need to calculate, treatment total and mean treatment. So these you can easily calculate using simple calculator. These are summation only. So second is degree of freedom. You need to decide the degree of freedom. So for total number of observation, there are 20 observation. There are 4 replicate value, and there are 5 treatment. So there are about 20 observation. So total 20 observation are there. So 20 minus 1 is 19 degree of freedom. Total degree of freedom is 19. Then block degree of freedom. That means replicate degree of freedom. So there are 4 replicates minus 1. That means degree of freedom is 3 for replicate. Then treatment degree of freedom. So treatment, there are 5 treatments minus 1, that is 4. So 4 treatment degree of freedom. Then error degree of freedom. So formula for error degree of freedom is total degree of freedom minus block degree of freedom minus treatment degree of freedom. So total degree of freedom is 19 minus 3 and minus 4, that is 12. Alternatively, you can multiply the block degree of freedom and treatment degree of freedom. So 3 multiplied by 4 gives you the same error degree of freedom. So there are 2 ways of calculating the error degree of freedom. So now we have decided the degree of freedom. Next step is calculate the correction factor. So this is third step is very long. We need to calculate the grand total square and divide it using total number of observation. So Ex summation square divided by total number of degree observation is 20. So this is the value 845 which we have observed here. Total grand total. So grand total divided by total number of observation gives you the correction factor. Next is calculation of various sum square. So this step is very long. First we need to calculate the total sum square, then replicate sum square, then treatment sum square, then error sum square. So total sum square is Ex square, these individual x values are there, minus correction factor. So individual value 15, 17, all the individual value in this table, 15, 17, 16, 18, up to 88. All you need to add. Before adding, you need to make square of these value and then subtract the correction factor. So you get the total sum square. Next is block sum square. That means replicate sum square. So we have a block total. So there are 4 block totals, which we have earlier calculated. This is 204, 209, 220, and 212. So 204, 209, 220, and 212, we have made a square and then summation and then divided by total treatment minus correction factor. So this is how we can calculate the block sum square. Similarly, treatment sum square. So treatment total square summation and then divide by number of replicates. So in this way, you can calculate the treatment sum square. Then error sum square. So error sum square is total sum square minus block sum square minus treatment sum square. So this is how you can calculate the error sum square. So this is the third step completed here. Fourth step is dividing each sum square by its corresponding degree of freedom to obtain the mean square value. So mean square value for the block is block sum square divided by degree of freedom of your replicate. Similarly, treatment mean square, error mean square also you can obtain. So mean square value are you are calculating by dividing the sum square divided by degree of freedom. So now you can easily calculate the F value. So treatment mean square divided by error mean square gives you the value of F or treatment. So F value for the treatment is 229.61. So block F value, block mean square divided by error mean square is your block F value. So as you can see, within the replicate, the F value is very less, but between the control and treatment, the F value is very high. We can place this our value in our result table. So here, degree of freedom, sum square, mean square, and observed F value we have placed. Now we need to compare this observed F value by the tabular F value. So table you can easily find out in any statistical book. We have a critical F value, and using the degree of freedom you can place over tabular F value. Now you need to compare the observed F value with the tabular F value, and for comparison you have some rules. So if our observed F value is greater than the tabular F value at 5%, so variation is very significant with 95% of probability. So such result we can denote using single star. If observed F value is greater than the tabular F value at 1%, that means the variation cause is highly significant with the 99% probability. That means our error is less than 1%. And such result we can denote using double star. If the observed F value is less than tabular F value at 5%, that means the variation cause is non-significant with 95% of probability, and we denote such result using NS. So this is our final result. The F value for the treatment is 229, which is significant in comparison to the tabular F value. And F value for the replicate is non-significant. So our earlier aim was to estimate the value of ANOVA and to determine whether there is a significant difference due to treatment or not. And in this example, we can see that yes, there is a highly significant difference due to treatment. So the same ANOVA, the manual calculation we can calculate using Prism software also. We will have a look of the Prism software also. First, we will calculate using some Excel sheet. So I am sharing the screen for the Excel sheet now to determine the correlation and regression. So suppose this is, I will delete these everything. So this is our treatment. We have given 0 milligram zebralic acid, 10 milligram, 20 milligram, 30 milligram. And these are the 3 replicates. And we are measuring the plant height after 1 month. So this is the plant height in centimeter. This is known as response. So now we want to estimate the mean value. So as you can see on the Excel here, A, B, C, D, E, F is written, and here 1, 2, 3, 4, 5 is written. So to identify any cell, we can easily say that F2 is this cell or G2 is this cell. So now we want to calculate the mean value for these 3 replicates. So we will select this particular cell and we will go for the insert function. And in insert function, if required parameter is not here in most recently used tab, then we can go for the all button. Otherwise, so we will select the average for calculation of the mean and click on OK. So by default, Excel has selected the nearby cell B2 also is selected and taken as a replicate value, which is a wrong. So we need to calculate the mean for these 3 replicates. So we need to select only these 3 cells. So we need to again select these cells. So C2 to E2, that means from this to this are 3 replicates. So we have calculated, we need to select these 3 values and we will click on enter button and click on OK. So this is the replicate value for these 3 replicates. We can drag the Excel, this particular cell, and we can copy paste the formula for below mentioned value, and this will give you the mean value for the rest of the values. Now we want to calculate the standard deviation. So we will select this particular set and go for the insert function and click on the standard deviation, click on OK. So again, the Excel has selected B2 cell because B2 is taking, this Excel is taking as a replicate value, which is not true. So we will select again the replicate value. We will click on this arrow and select the replicate value and click on enter and click on OK. So this is the standard deviation for these 3 replicate. We will drag down the cell to calculate the standard deviation for rest of the values. Now we want to make the graph. So we can go for the insert function and select any particular graph of your choice. Suppose I have selected this. Now this is a blank graph. I will have a right click on this and select the data and add the series. So I want to make a graph of mean value. So this is the plant height. I will give a name, plant height, and series value I will select mean value. So this is selected. Now I will click on OK. So this is the graph. As you can see that X value by default, the Excel has taken 1, 2, 3, 4, which is wrong. So we need to place or correct X value. So I will go for the editing of the X value and I will select these correct X value. So now the correct X value are mentioned here. There is no title mentioned in this particular graph. So if we go for the publication, it will be rejected. So we need to identify the label. So we will go for the design. After selecting this graph, it will show the design. Add the axis title. So primary horizontal title, you can mention here as a treatment. Suppose in milligram I have given. So it is better to estimate the unit also. So treatment in milligram per liter. Now the same graph I will go and add the vertical title. So this is the vertical title. So this is your plant height and in centimeter. So you can select. I can make a bold and I can place this plant height little away. So in this way, I can make a graph. In this graph, we have not yet mentioned the standard deviation. So it is better to mention the standard deviation. Go to the add chart element and go to the error bar option. on error bar option, it will show more error bar option. So we will click the more error bar option here on this side. We need to add our value, which we have calculated. So customized value we will select and click on specify value, and we will enter our value, positive error value and negative error value, and click on OK. So this is how the graph is showing the error value also, standard deviation value also. So in this way, it is better to represent the graph in this way with the standard error value also, so that publication is very easy, because this shows that you have done the experiment 3 times and you have calculated the standard deviation of those 3 replicate value. So it is easier to convince the editor of the journal that yes, you have done experiment in proper way. Now we can calculate the correlation value, whether the treatment and mean value are correlated or not. So we can select this particular cell and go for the insert function. I will go for the Pearson value, and Pearson, one array is your mean and another array is your treatments. So these 2 values you have calculated. So R value is 0.56. Now we can go for the regression analysis of this particular graph. So I will go for the again design option and add the trend line option. So I can select either the linear or some more other option. So in this demo version, I am going with the linear. Otherwise, you can go for the determination of the R square value, and the line which fit best, which gives you maximum R value, is the best fit. So I have selected this linear line. I will go for the format trend line after selecting this linear line, and I will display the equation. Equation means regression equation on the chart. So I will click on the display equation, and this is your regression equation. So now I can place this regression equation here and increase the font size. So your y is equal to a plus bx. So a value is your 12 and b is 2.9. So in this way, you can calculate the regression, you can calculate the correlation, mean and standard deviation. Now I will close this Excel sheet and open the Prism software for the ANOVA analysis. So this is the Prism. As soon as you open the Prism, a window will pop up. In this window, you need to select a single column of value. So I will click on a single column of value and click on OK. So that this empty Excel sheet will be appeared on this software. Now I can place my X value and Y value on this Excel sheet. So I will open the data. This is the data table. So this is the same data which I have mentioned in the my presentation. So these are the X value. I will copy X value here, and I will paste the replicate value here, copy and paste here replicate value. So these are the 4 replicates and these are the 5 treatment. Now I can click on analyze button and click on one way ANOVA, OK. And because we have a different replicate also, so I will check the repeated measure test because we have 4 replicates and click on OK. So this is the final result of our ANOVA. The replicate summary is non-significant and the F value for the replicate is 0.658. It is the same which we have estimated manually. Treatment F value is 229, which is same, and summary is that your treatment is having highly significant difference. And in ANOVA table here, you can find out the sum square between the treatment replicates and degree of freedom and mean square of the treatment replicates also. So this is how you can calculate the ANOVA using PRISM software. So these are the steps. First, you open the PRISM software and you feed the values and click on Analyze button and select the one-way ANOVA and check on the repeated measure and click on OK to get the results of ANOVA. So this is all in the presentation. We have learned the basic concept of correlation and regression, and we have learned the type of error, degree of freedom, level of significance, and ANOVA. If you have any doubt or you need this software, you can easily contact me. I'll provide the software to the organizers. Thank you so much for patiently.
Thank you so much, sir, for excellent presentation. Sir, there is a request from Dr. Mrinali Chauhan, ma'am.
Yeah.
She is asking, your lecture is very useful. Please provide us its recording or upload on YouTube and provide us its link.
Sorry, YouTube is available. This entire presentation is going live on the YouTube also, so you can give the link to that, madam. And anything else?
And ma'am, this lecture is already recorded on YouTube. Okay. Okay. Thank you so much, sir, again. Sir, you have made even the most complex concepts so understandable. You have discussed basics of correlation, regression, and ANOVA with so simple examples. Especially the way you have explained degree of freedom was so understandable. Uh, as I know him personally, apart from being a good academician, sir is a very affectionate and a very wonderful human being. He has a positive and pleasing personality. Sir is always ready to mentor students, and I have also learned a lot from him. Sir, on behalf of Directorate of College Education and Government College Bundi, thank you so much for your excellent presentation. Thank you so much.
Thank you. Thank you. Thank you, Nikita.
थैंक यू हरी सर। सभी प्रतिभागियों से आग्रह है कि फीडबैक आप लोग भरें। अब इसमें से मैं कुछ नाम बोल रहा हूं जो कंसिस्टेंटली फीडबैक नहीं भर रहे हैं। नवीन जी, महेश मीणा जी, रमेश चंद मीणा जी, इसके अलावा महेश चंद मीणा जी करौली और रमेश चंद मीणा जी दौसा। नेक्स्ट है अनीता मालव, राजकीय कन्या महाविद्यालय कोटा। मधु चौधरी, राजकीय महाविद्यालय लालसोट। आप सभी से निवेदन है कि आप कृपया फीडबैक दें हमें। बार बार ग्रुप में भी हमें लिखना पड़ता है, अच्छा नहीं लगता। आप सभी बड़े हैं, अग्रज हैं, सम्मानीय हैं। आपका ये फीडबैक रिपोर्ट जो